Hi. So my name is Grace Coppinger. I'm from the University of South Alabama, and actually there I study venom transcriptomics in garter snakes. But I wanted to do an REU to get kind of a different aspect and a different side of the research world because my university is relatively small. So this was my opportunity to do that.
Oh, that's awesome. Thank you. Would you be able to tell us like a bit about the awesome research you've been doing as well? I can
Absolutely. Okay, sure. Yeah. So I wanted to focus on two things. One of them being watermelon and then the other being structural variants. Watermelon is one of the top 5 most consumed fruits worldwide. And because of that, there's been a lot of domesticated changes and phenotypic changes. So the cultivated watermelon that you typically see in the grocery store is super sweet and super soft. And then the wild watermelon that you don't see in the grocery store, obviously, is very hard, tough, and not tasty to eat. So knowing this, we also wanted to look at structural variants, which are deletions, insertions, translocations, inversions that cause a large amount of the genetic genome to change. So that's what we did. We did a whole genome alignment, and then we identified structural variants. Yeah, it was a lot of numbers. We identified all the structural variants within the genome. This is kind of a table summarizing the amount of insertions, deletions we found, and then how we identified structural variants to kind of annotate going forward. And then after we got this list, we were like, wow, this is a lot of genes that are affected. Let's do a differential expression analysis to kind of decide which genes are also affected by, or which genes are also being expressed during fruit development. So then we did a side-by-side comparison, and we ended up having a list of 87 genes that are both differentially expressed during fruit development and affected by structural variant within the cultivated genome, and then 192 in the wild genome. Yeah, so in future directions, we're hoping to look at disease resistance because during this project, we found 7 different genes that were involved with disease resistance and also affected by a structural variant. And the wild genome is resistant to fusarium wilt. The cultivated genome is not. So this is something that we can go on to do in the future. So I talked really fast, but
yeah, it's okay. I do the same. No worries. Wow, this is such fascinating. Yeah, so much work. You've been working hard this summer. I just have a question. So, you know, in our cultivated watermelons, like a lot of sweetness, the color differential, and then you mentioned in the wild, we don't see, or in the wild, we see this disease resilience and not in the cultivated. Do you think that there could be any linkage between resilience and some of the color and sweetness flavors that we see in our cultivated?
Yeah, there definitely could be. I didn't necessarily look at that in my study, but that is something that could definitely be related. I think doing more gene annotation and gene analysis studies would be the answer to find that out. Yeah. Yeah, sorry.
No worries, gosh. Yeah, it's oh wow, this is such fascinating work you've been up to. Is there anything that you found surprising about the differentially expressed genes within the cultivated and wild that you weren't necessarily expecting?
Sure. So this graph is my favorite graph. This is a Go enrichment analysis that we did, and genes that are highlighted in here are functions that were found in both the cultivated and then wild genome. And they're genes that are being expressed during fruit development. And then highlighted in red are actually genes that are related to fruit development only found in the cultivar genome. So just looking at this, you can see how far we've domesticated this plant species and how much more or much higher expressed that these genes that are directly related to fruit development are. But yeah, sorry.
No worries. Oh gosh, don't be sorry. I look forward to the day that we have cultivated watermelons that are disease resistant.